So, good afternoon, everyone. So now I'm now NISHIDA Nao, a third-year assistant professor at WIAS, and thank you very much for joining us today. And today I'd like to talk about my research on how cancer EVs are transferred within the tumor tissues and incorporated into the target cells. So cancer is currently a top cause of disease in death in Japan, and almost half of the people are contracted by cancer in their lifetime. So it is a huge issue in Japan and also the worldwide as well. So tumors arise in many locations in our body, but the tumor is this kind of mass is not only a mass of the cancer cells, but also it consists of a variety of the infiltrated non-cancerous cells, such as immune cells or endothelial cells and fibroblasts. And also there are non-cellular scaffolding components like collagens and other extracellular matrix. And cancer cells interact with these surrounding non-cancerous cells through direct contact and also the secretory factors, including extracellular vesicles, which I am studying, to modulate them into the tumor supportive phenotypes to establish the tumor microenvironment, which support for cancer growth. And extracellular vesicles, or EVs, are nano-sized bubbles like this shape, round shape, with almost 100 nanometers to 5 micrometers in diameter. And this is a lipid bilayer vesicles. And within these vesicles, they contain cellular components like proteins or RNAs or DNAs. And the EVs are secreted from one cells and uptaken to another cells and release their contents to cause changes in their recipient cells. And this is how the EVs transmit intercellular signals from one cells to another cells. And cancer cells secrete lots of EVs and utilize them to modulate the surrounding non-cancerous cells to help the cancer cells to grow. Further, cancer cells secreted EVs, and the EVs travel systemically via blood vessels, bloodstream, or lymphatic flow to reach to the distal organs and alter the environment to a favorable environment to support the cancer cells metastasize and grow at the distal organs. And after reaching to the distal organs, the cancer cells further secrete the EVs to modulate the surrounding cells to establish more tumor favorable environment. And in my previous study, I actually blocked the cancer EVs using the antibodies and to show the therapeutic efficacy to suppress the lung metastasis. So my research question at WIAS is how the cancer EVs are transferred within these tumor tissues. So this is an electron microscopy image of the colon cancer patient tissues. And this shows one cell, and this shows another cell. And red arrows, I don't know if you can see here, but like the red arrows are all EVs secreted from these cells. And you can see that the EVs secreted towards this small intercellular space of these tightly packed tumors. And this is a schematics of the tumor tissues. And as this schematic shows that the tumor tissues are very highly packed structure with the variety of the non-cancerous cells, cancer cells and non-cancerous cells, and also the collagens and the scaffolding extracellular matrix. So, and the cancer EVs are secreted to this small interstitial space crowded by this variety of the cells and structure. So under this kind of conditions, there are several questions arise. So first of all, like which cells in these tumor tissues will uptake the cancer derived EVs, or like how far the EVs that secreted from one single cell, cancer cells, can reach within this highly crowded structure, or how fast they can move through this structure. So, but however, these kind of questions remain unknown because currently the EVs are mainly studied in cell culture-based research or like animal studies. And these systems are kind of lacking, cell culture systems are lacking the physiological tissue environment. And in animals experiment is kind of technically challenging to observe the EV transfer cellular levels due to their size, the nano size. So to truly understand the cancer EV transfer and resulting cancer cell to the surrounding cell communications in real tumor tissues, we really need the tissue-based experimental system, which allows the EV observation and the further EV studies. To overcome these technical difficulties, I am currently utilizing the technique called the tissue slice culture, which I will explain later. And by combining with the highly sensitive EV levels, I will be able to observe the cancer EVs transferred directly in the intact tissue architecture. And currently, I am using the breast cancer as a main cancer model, and I am using this system, I am addressing these tissue-oriented questions I mentioned earlier, like which cells are uptaking the EVs, or like how far, or like how fast. And also I'm trying to determine the tissue factors and other factors that are defining these tissue EV transfer patterns. So what is tissue slice culture? So tissue slice culture is a thinly sliced tissue that maintains life in the lab with the culture medium like this. And this tissue slice culture is just prepared, maintains the original tissue architecture and the cellular profiles, and can be utilized as the tissue models for many experiments, including the EV observations. And this tissue slice culture can be prepared from basically any kind of solid tissues like tumors, or other organs, and also patient tissues as well. And this is actual movies of the tissue slice preparations. And this using the vibratome, tissues are soaked, located at the center on the stage and soaked in the buffer, ice cold buffer. And using this machine called vibratome, the tissues are sliced into this 215 micrometer thickness. And this size is collected and further cultured in the lab. And I have prepared the tumor tissue slice culture, but here I have optimized the conditions to prepare and maintain the tissue slice cultures from other like mouse organs, like brains and lungs and livers, where the breast cancer I am studying is likely to metastasize. And I have confirmed that these tissue slice cultures maintain the original tissue architecture, like here, brain, and also the liver. They have the unique structures. They are maintained here. To first check whether I can use this tissue slice culture for EV observations, I first supplemented the EVs, collected and isolated from the breast cancer cells, and I stained the EVs with the PK dye, green fluorescent dye called the PK67, and supplemented to this tissue slice culture of the breast tumor slices or lung slices or liver slices. And here the cell nucleus are all stained in blue, DAPI. So all the cells, including cancer cells or other cells on the tissue size cultures, are stained in blue, and the EVs are labeled in green. So you can see that some of the cells on the tissue size cultures are actually incorporating the supplemented EVs and shows the green signals. And especially, you can see that the uptaking pattern is not uneven. So not all the cells on the tissue size culture are incorporating EVs, cancer-derived EVs, but some of the cells are preferentially uptaking the EVs. So this image shows. So to observe the cancer cells secreting EVs and transferring to the non-cancer cells at the more physiological conditions, and especially I focus on the metastatic conditions. I first established the ex vivo breast cancer metastatic models. This time I have optimized the lung metastatic models and brain metastatic models. Here's the brain metastasis model results. And I have prepared the mouse brain slice here and supplemented these breast cancer cells sparsely onto this brain slice and culture them. Then I was able to see these seeded cancer cells integrate and invaded inside of this brain tissue slice. And here in the cancer cells, I introduced the highly sensitive EV levels in these breast cancer cells, which are prepared by the overexpressing the halo-tag fused CD63, which is a protein enriched on this EV. And also I have introduced the nucleus CFP as a level for cancer. And so using this model, we have observed the EV transfer directly onto this brain metastatic models under we performed the live imaging using the 2-photon microscopy. And this kind of for establishing this brain metastatic models like preparations and seeding conditions, culture conditions, and also the observation conditions, all kinds of optimizations are thankfully performed by Dr. Hatashita, former PhD students at Dr. Inoue's lab. So this is the live imaging of this brain cancer cells secreting these breast EVs on this brain metastatic models. And this is cancer cells. And you can see that like small dots are secreted from these cancer cells towards the outside. So this is how the cancer cells are secreting EVs towards the surrounding environment. And these EVs should be incorporated to some of the cells on this brain slice. Okay, so I have prepared the brain metastatic slice models, but here also we have prepared the brain metastatic models using the highly metastatic breast cancer cell line called BMD2A, established from the original MDMD231 cell line. And this one cell line because it's highly metastatic, I expect that they are secreting more EVs and transferring more EVs towards the surrounding cells. But these images are taken by Ms. Moe Saito, the undergraduate at the Dr. Inoue's lab, thankfully. And yeah, here from now, now I have just we have just established this model, but from now we will just observing carefully and compare the results with the original cell line, whether they have different secreting or transferring patterns. And here is a result of observations of the lung metastatic models. It is the same. Basically, it's the same. So the cancer cells level with the high sensitive EV levels are introduced on the lung tissue slices to prepare the lung metastatic models. And this time, I have observed under the confocal microscopy is a static image. And then here the EVs are labeled with yellow, and then cancer cells has a nucleus CFP here, and all the other like cells shown in blue nucleus patterns are from the lung slice. And you can see that here's a lot cancer cells located here, but there are several EVs surround, and these cells from the lung slices are preferentially incorporating this cancer-derived EVs. So currently, I am trying to identify which cell types are preferentially incorporating the cancer EVs here. So what are these cells? So like how I can identify which kind of cells. So currently I am trying to perform this. I decided to analyze comprehensively using the flow cytometry. I first prepared the lung metastatic mouse breast cancer models by injecting the cancer cells to the mice, and they developed the lung metastasis. Then I collected the lungs and dissociated the lungs into the single cells and analyzed all the cells. And here the cancer cells have the EV levels. So I can identify which cells are really incorporating these cancer-derived EVs. And this is a very preliminary result, which I was able to obtain recently. And you can see that among the variety of the cells in the lungs, you can see that some fibroblasts or lung epithelial cells are maybe more than 20% to 45% of these cells are actually incorporating these cancer-derived EVs, whereas these kind of immune cells like T cells or B cells or myeloids or other blood vessels derived cells are not really preferentially taking. So it's only about 3% are positive with cancer-derived EVs. So this result explicitly shows that the EV uptake is like happening preferentially among the different cell types, and EV have definitely have the specific preference in incorporating into these fibroblasts or lung epithelial cells. So for now, I have the main result I have is so far that's all. And to summarize, I am leveraging the tissue slide culture platform and directly observe the cancer EV transfer within this intact tissues architecture. And by combining this kind of cutting-edge analysis, I'll be able to observe the EV transfer directly in real time and identify the target cells and spatial distribution of the EVs, and eventually I'll be able to understand the cancer cells and other cell communications networks within the tumor tissues. And this kind of experiment eventually leads to the developing the novel therapies for this targeting this kind of cellular interactions. Future plans are I am trying to identify the cell types and analyze the kinetics of the EV transfer, the speed, and the spatial distributions. And they also compare the distributions among the different highly metastatic brain, highly metastatic cancer cells and less metastatic breast cancer cells to understand the mechanisms of the metastasis. So lastly, I'd like to thank all the members, also the members of the Innovate Lab especially. And I know it's overtime, but like I just want to add several things a little bit. I here like WIAS, I was able to I was very happy, lucky to start several collaborations. One is this collaborations, which I get to know Dr. Akimitsu Narita by Jahoe 2023, which Dr. Tokoro Sensei recommended me to allow me to join. And he was a chemist to develop the new dye to level the EVs, and I'm leveling his new dyes with EVs. And also he has a collaborators at Max Planck, Dr. Liu. He for specialties is in microscope, high resolution, super resolution microscopy, and we are collaborating to observe the single EVs under her microscope. And I also got these unexpected findings, and the project are really expanding right now. And also I was able to start a new collaboration with Dr. Alain Hang. I got to know at the International Conference recently. He will be helping me to analyze all the changes of these surrounding cells in surrounding phenotypes. And he came to the twins recently and gave the lecture as a WIAS Top Runners lecture series. And I also have several collaborations. Other projects like Dr. Takao Yasui and also Dr. Yamamoto at National Cancer Center. And also recently I started this Takeda pharmaceutical collaborations as well. And there are also other interesting research going, but unfortunately don't have time to introduce. So I'll skip these kind of things, and thank you very much for attention. Thank you.